Okay, well, I'll assume that Mary will get this done by the time we get the introductions done, because I'm gonna actually introduce the person who's going to introduce Mary. So I first just want to welcome you. This is not only the regularly scheduled Thursday afternoon Ecology and Evolution seminar, it's a seminar that is getting support as a Storer lecture, and we owe a debt of gratitude to the Storers for providing the endowment. And I am going to hand over the mic to Tom Shaner to introduce Mary, and I think you'll hear why Tom is doing the introduction.
Mary was my graduate student. Is that a good enough reason why? So I've got her CV, and she got her BA from Brown, her MS from Boston University, her PhD from the University of Washington, also with Bob Payne, on grazing armored catfish in Panama. And then she got into the Eel River, and had never really gotten out, as far as I can see. And among her she's written a lot of classic papers, but her paper on a 4-level food web, the trophic cascade, is, it was an instant classic, and now it's an older classic, but it will never go away as a classic. It is probably the most important paper written about simple food webs that I can think of. Accolades. So there's 2 pages of those. She has been assistant associate and regular professor in integrative biology at Berkeley. Most of you know. She got a Guggenheim. She got a Miller Institute for Basic Research professorship. She got the G.E. Hutchinson Medal from the American Society of Limnology and Oceanography. She was the president of the American Society of Naturalists. She was the president of the Ecological Society of America. She's a fellow of the American Academy of Arts and Sciences and a fellow of the National Academy of Sciences in the U.S. And there's a lot more, but we don't have time. So I'll just conclude really quickly, and I'm not quite sure what I'm going to say here. We all owe her a huge amount of appreciation for all those accolades, much of which is service. We have much admiration for her pioneering research. Absolutely innovative, path-breaking, and original. And most of all, personally, much affection. Both, it was a great privilege to have her as a graduate student, and it's a continuing fantastic privilege to have her as a colleague.
Thank you so much, Tom. Button. Well, how lucky was I to meet Tom when he was just about to leave Harvard and come to University of Washington. And I think our interview was just chatting about the natural history of cenotes the whole hour, the sinkholes in Mexico, where I wanted to work but couldn't quite figure that out. And then what a privilege to get to work with both Tom and Bob. I couldn't have been luckier. And to have 3 years to live in Panama, trying to figure tropical streams out. That's what we're all in it for. So let me just, I have way too many slides, so let me just, let's see, I think I need to do this. So we're all, I think many of us in the room anyway, are in this initially because we want to get to hang around with organisms and figure out how they make a living out there. How do they get enough to eat? How do they avoid being eaten in this really occasionally dangerous and sometimes very rewarding environment they live in? But how do they deal with the fluctuations? And when you get into the food web business, you get very interested in what limits a node in that web, otherwise known as an organism. So I think what distinguishes Tom and Bob Payne and many of us in the room is that we think of these as organisms that have rich natural histories that we really care about. So you can't look at a lot of them. And so we tend to look at just a few at a time and just cross fingers that other dynamics will be entrained to whatever happens to those few. So you have to make a lucky, and I won't say judicious, I'll just say a lucky choice of what few. And we know that so that what limits them, consumers of resources, is going to change through time, especially in the acceleratingly changing world that we live in. So how are they going to change, and how are their ecosystems going to respond to the ways that they change under forcing? So I know you all are keenly aware and motivated by climate change. How many of you have seen this thing by Gregory C. Johnson? He illustrated the last IPCC report in 19 illustrated haiku, which he illustrated with his own watercolors. And here's one of my favorites. This is online, so you can get it. Wet will get wetter and dry drier since warm air carries more water. So they're all beautiful haikus, and they distill it down. And so as Tom said, I did have a sojourn into the Ozarks and prairie streams for a while, but then I found the North Coast and have basically committed my last 30 years to trying to figure it out. Also, it's protected. There's a protected research site, which the UC system's so fortunate to have our natural reserves, and I've been very fortunate, as Sharon Lawler also said, and Susan Harrison, our research is empowered and protected by having these protected research sites. So thank you, Angelo Reserve. So Angela sits in the North Coast, which California, especially Southern Californians, consider a very water-rich area, and they had all sorts of nefarious schemes for draining the eel south. But sometimes that's true, and we get big floods when we get these atmospheric rivers that shape up near Hawaii and bring warm air swirling together with cold air masses, and they slam into the West Coast, and that's what we've experienced last week. And in 64 and 55, we had really big ones that wiped out a few towns and bridges along the eel. So it was just shortly after that that under the Carter administration, they were thinking of sucking the eel south, but a heroic man, a single rancher, initially stopped that. And that hasn't happened, though, as much recently, and we all know that we've had this really devastating drought from 2013 to 2016 that affected California, set up by the mysterious blob, the warm air mass that accumulated off our shores and diverted all of the atmospheric rivers and the warm air to heat up Alaska and then to go down and drench the Midwest as we died of thirst here. And just recently, there was a nice summary article in February 1st Science on figuring out what happened to make that blob. I understand at the time it took the climate scientists a while to figure out why we were having this persistent warm air mass, but somebody has at least traced it to warm air setting up in the Gulf of Alaska and then spreading south to cover a huge amount of the ocean. And the alarming thing that I learned in this little review is that another one's shaping up. There's a baby blob shaping up. Warm air is settling in the Gulf of Alaska, so we've got to keep our eye on the future and see if this happens again. During this drought, the Eel River, which drains over 9,500 square kilometers, went to ground for the first time in recorded history, written history. And this is what it looks like going to ground quite near its mouth. So it happened in September, and folks thought, well, that's at least okay for the fish because the smolts have gotten out and the incoming adults won't get stranded upstream somewhere and cooked to death. So they thought, well, if it had to happen, at least it's happening in September. But what you can also worry about is what's going on with the rest of the salmon-bearing food web, or the rest of the food web if you care about other organisms. And so I want to emphasize today on what high flows and low flows do to attached algae that are supporting most of the production, most of the animal growth in the whole river network. I'll justify that shortly, but that claim, until you get to the estuary where we don't know what's going on. At least I don't. So before we go into algae, notice I drew this green heart around the algae, hoping to start to get you to care about them and their lives. So we do have what Clint Eastwood might call the good, the bad, and the structural. So the diatoms are what you see initially when stones have been scraped bare, early succession. And then if there's no abrasion and no grazing for a long enough time, like weeks, you tend to get these long filamentous chlorophytes. So just like a forest recovering from a fire, light gets more limiting, and the tall algae have an advantage if they're not grazed and if they're not scoured. So these then also provide a secondary substrate besides stones for diatoms. So you get epiphytic diatoms covering this green stuff, and you'll see pictures of them, and epilithic diatoms on the stones. And then over time, another thing can happen, which is this assemblage of Cladophora, the green alga, and its diatoms can get studded and then blanketed by cyanobacteria. So how many have heard about the dog deaths in the eel in the Russian rivers? So that's gotten our neighbors' attention and ours, of course. But before we talk about that, let's just talk about the fact that these assemblages of algae are something that we've kind of seen worldwide in the exposure that Ivan Vadabankor has gotten in the eastern North America and in Africa with looking at Mbuna feeding cichlids. What I've seen in the Ozarks and prairies in Panama and in the eel is 3 basic assemblages. You get diatom skins with certain kinds of chemistries and substrates if it's heavily grazed or recently scoured. You get cyanobacterial felts, not drawn to scale here, but felts that are less than 1 millimeter high but look velvety. And both of these things are renewing like crazy, and at least some of them are highly nutritious, so they can support tremendously inverted pyramids of consumer biomass, lots of fish, in other words, in Panama, in Africa, and lots of herbivorous insects where fish aren't important grazers. If the algae has been released from grazing long enough, it grows into this turf, and then it gets infested by not only epiphytes but bugs, and you'll see some consequences of that for the eel river food web where grazing fishes are not having a huge effect. But just quickly, here's what it looks like. Here's Mbuna cichlids keeping this cyanobacterial felt really grazed down. If you remove them, you get clumps, and this is a predatory fish in Lake Tanganyika. And then here's the Ozarks, where you have black velvet felts of cyanobacteria that are velvety, and you remove the natural or the artificial substrates from grazing, they get turfs of diatoms. So you put that stuff back in the river, it gets scoured off immediately, and black felts regrow under chronic scouring. So grazers are really controlling these, and here's a place where, as we'll talk about shortly, floods, wet winter hydrology has released the algae from grazing for long enough to give it a head start, and you get the chlorophyte assemblages. But if you have floods, then you might get these big, huge growths for both bottom-up and top-down reasons. Bottom up and top-down reasons. And because, watershed like the eel, if you have a wet winter, it'll drive more nutrients into the water for the summer, and it also, higher summer flows will prolong good growth conditions. So those are both bottom-up reasons for getting a big algae year after a wet winter. Also, there's the hypothesis that we've found a lot of support for, that you're extirpating big caddisflies that by early summer grow too large for fish to control. So that's the top-down hypothesis. So how do you evaluate their strengths? I'm going to do that at 4 watershed scales, very tiny watershed down to the whole basin, actually. We could actually probably use some collaboration with all the smart numerical minds here in terms of really quantifying the relative strengths of bottom-up and top-down forces in determining algal biomass. But we have convincing evidence, I think, that top-down is important at a lot of surprising scales. And one of them that's really surprising is starting up in the dark headwaters. So the resource that gets really limiting as you go into narrow, small headwaters is light. And so we'll start out thinking about what's going on for algae and grazers up in the tributary tips of the headwaters of this area. This is the area around the Angelo Reserve, which is the upper drainage of the South Fork. The Eel drains north. And then we'll start looking at larger and larger scales, larger tributaries, main stems, and then down to the whole basin scale here towards the end of the talk for this river network. Basically, the trend is very predictable, and if any of you are interested in these factors, theoretically, you might consider river systems as a system to study in the field, because you have a quite predictable gradient of, in the headwater tips, you're resource limited, so you grow slowly, but you're not as subject to disturbance. The bed gets disturbed here every century or every millennia because it's just debris flows, which are rare at any reach of river. But you're growing very, very slowly. Down here, you're growing much, much faster, but you get disturbed on average every year, every other year by bed scour. And if you get down to the mouth, you're chronically disturbed. So there's this wonderful gradient of productivity, how fast can you grow, and disturbance, how long do you have to grow before resets to look at. Now, the reason we know that algae, benthic algae, attached algae, is the base of most of the food webs here is the work of Jacques Finlay, former graduate student who's been a professor for a while now at University of Minnesota. And Jacques used carbon mostly in his thesis work, then he's collaborated to use fancier isotopes to really try to nail down how much is allochthonous input, how much is allochthonous is a terrible Greek word that I shouldn't have used. It means leaves that fall into streams. In this case, it means it comes from outside the habitat. But how much is twigs and leaves, litter, versus algae growing on surfaces in the water? And so here's about as far up in the drainage area as you can collect insects. It's drains about 0.5 square kilometers. And you can't detect algae. We humans can't detect algae, but caddisflies can, and some of the caddisflies, the Glossosoma, for those of you who know bugs, have carbon signatures that look like they're entirely built of algae, even up here. However, they're not suppressing algae. I'll show you those data shortly, but if you remove caddisflies or protect algae, you don't get algal increase. Is this microphone okay? It seems like it's okay. So here's the picture of the food web in this little tiny drainage. These caddisflies are built of algae, but all of the mayflies and midges and the invertebrate stoneflies that eat them and the salamanders have a terrestrial carbon signature. So you go then down more than double that drainage area, and it's still a pretty small looking stream, and it's still a stream that any stream ecologist or any naturalist would say, well, there are no algae here because it's light limited. However, Camilla McNeely, and the isotopes, though, tell us still caddisflies are eating it, and these guys are still largely built of leaves. But when Camilla McNeely took out thousands of tiny little stone-cased caddisflies per square meter, and she replicated that for 8 treatments, removal and controls up the stream, she released turfs of about 8 to 15 centimeters of highly edible diatoms that could tolerate and grow in low light if they weren't grazed. So that was one experiment, and then we did simpler, easier experiments. She used this elevated tile technique, but she improved it over, and this is from my work in Panama, and I don't have a photograph of her fancy setup, which was to take a plastic food container and put a stone in it and invert it, and then put the tile there, and the bugs can't crawl up the plastic container. So that works pretty well, especially with Vaseline. So here is the headwaters that we looked at before, and if you protect these grazing substrates or expose them, you don't get a difference until you get to 2 square kilometers of drainage area, and that's enough to have the grazers build up numbers so that they really can suppress algae. And then that continues down for the duration of the stream. The next tributary I'll talk about will be this 17 square kilometer tributary where the stream's widened and it has a half open canopy, but still the substrate generally looks quite barren. However, the isotopes that Jacques looked at are telling us that not only these armored caddisflies, but all the other invertebrates and the fish that are starting to show up here are built of algae largely. there's enough noise in the data that we can't say they're entirely algae, but they're probably way over half algae. And here's what happened in this stream. You probably know California well enough to know that our storms are big. We don't get those little patchy storms like they get in the Midwest or Texas. So when we get slammed on the North Coast, the whole watershed is either really inundated and flooded or not. However, one year, strangely, we got what I call a hydrologic gift. It was kind of a miracle. And it's that Elder Creek, that 17 square kilometer half-shaded basin, totally scoured. We got a patchy rainstorm that put it way up above bank full. Here are dates. This is the year I'm talking about, the water year I'm talking about, of 2010. And here's the bank full level at about 10 cubic meters per second in Elder Creek. So this was a scouring year, and for the only time in history that I've ever seen this, the South Fork, the main stem, didn't scour, didn't come close to bank full scour. So that was a gift that did the experiment for us on the large 17 square kilometer scale, not just the elevated tile or the removal scale. So here's this basin, and it turned green. We'd never seen this before in 30 years, but the Elder Creek got vividly green. And so when you find large armored caddisflies there, not in great numbers because they're a bit food limited, but when they got blasted out, there was enough light to really release Cladophora. And this is what from the same year, this is the South Fork, where we're out in the open. Usually the big blooms start here. They never did this year because the grazers had not been removed. So here's the data. I promised Peter I'd only have 3 data slides, so maybe just one more to go. Here's the number of dicosmicus, density versus the amount of Cladophora. You know, you usually plot these in the reverse with the plants versus the grazers, but here we're wondering if the grazers are controlling the plants. And so this is the South Fork eel that year, basically zero grazers, zero Cladophora, and variable numbers of grazers that had not been scoured out, up to 14 per a quarter square, up to about 50 to 100 per square meter, actually. And then the dark points are aggregated points from Elder Creek, where we got the big releases of Cladophora and very, very few dicosmicus. So here's a case where the light-driven gradient of primary production was actually reversed by this unusual scouring event showing top-down overrode this under these unusual flow conditions. So I'm going to go to the main stem now and go over some older work, but you're familiar with our Mediterranean climate that dries up the rivers in summer and scours them during normal winter flood years. Those things are both changing under climate change. And so if you get your scouring event, you knock all of the rock-bound biology off the rocks. The insects are gone, the algae are scoured down to so you can't feel them, but there are basal pads and there's some cells left. And so by June of a post-flood year, post-scour year, you get these long streamers. This Cladophora that is released by flooding can grow 10 meters long, 10, 15 meters long sometimes in record years, and it branches. So it's enormous increase in surface area. Cladophora, human hair size, branching, has rough cell walls, so it's a great colonization substrate for diatoms. And that's why the floating mats that are collecting in slackwater areas here are turning yellow. But in a year like this, by late summer, you do get the turfs that are still attached and the mats withered back to remnants that actually have an architecture that's produced in many years, not all, by infestations of these chironomids that weave themselves little retreats or tufts, live inside them, and function as a grazer. They eat mostly diatoms, as you'll see, but they break up the Cladophora and so act as an herbivore in terms of reducing its biomass. So way back when, after I spent a few years trying to avoid writing a animal care and use protocol, I gave up because I wasn't learning a lot of repeatable stuff by looking just at the base of the food web. So I finally did fish, no fish experiments, and Jennifer Nielsen, who some of you know, helped enormously with that, teaching me how to shock fish and move them around without killing them. So we made these fish, no fish enclosures that Tom mentioned, and that led to that 1990 paper he was kind enough to mention, which is if you remove fish, surprisingly, you get very barren rocks within 5 weeks, 5 or 6 weeks. Here's a fish about 6 centimeters long. Here's a little centrifuge tube we use as sediment traps, about 10 centimeters high. The turf that had begun about 40 to 60 centimeters long had been grazed back or collapsed back to a little mat that was no more than 1 or 2 centimeters high. And so that was a very powerful, effective fish, which surprised me because I'd used midges in bondage experiment. This little midge that is creating all the little lumps there was heavily eaten by fish when they were withdrawn from their tufts, tied with a cotton thread and dangled in the river to see what's the landscape of fear like for you, little midge? So where is the predation intensity, high or low? And so fish love them, but they don't recognize them when they're in their little tufts. But it turns out invertebrate predators do. So invertebrate predators, of course, are eaten very rapidly by fish. They're big and clumsy and slow. But if they're not, then they stay abundant and they wipe out that midge and release algae to persist. So the algae where fish were absent, but those small grazers, not only these odonates, but also fish fry, anything that could get through a 6 millimeter mesh here. Those fish fry were abundant. It was a 3-level food chain, and this algae here went from bright green to scuzzy yellow color, and also these blobs showed up. They both turn out to be nitrogen fixers. The blobs are a cyanobacteria, nostoc, that fixes atmospheric nitrogen. The diatoms you'll learn more about shortly. Some of them include endosymbiotic cyanos, and both of them are doing what Hairston Smith-Watkins said they should do in a 3-level food chain, which is they're resource limited, so they're beating out the cladophora by fixing the limiting resource, nitrogen. So here, Tim Wooten and I, let's see, I should mention before we leave this that we got that result in 1989. The next year, pre-tenure, it was totally not obtainable. I built 24 enclosures, doubled the size because I was going to look at species differences in the fish, and the fish had absolutely no effect, 0. And then that happened in 1991 also. And then in 93, we repeated in 97. We got a 3-level food chain where fish had a big dynamic effect again on algae, but the midges didn't recruit. So it was a 3-level effect. They were actually protective. And in 1997, we had a 4-level effect too, where fish were bad for algae again. What was going on in 1990 and 1991? It was in the second year that we were smart enough to look around and notice that that's when the drought effects started showing up on the Eel River. And so Tim Wooten and I did the Wright experiment, which was to look at the big invulnerable grazer that after a flood, you'd find a couple of them per square meter. After a drought with no winter scour, you find 50-ish, sometimes 100 per square meter. They're patchy, but they're a lot more abundant, maybe 2 orders of magnitude more abundant if they haven't been scoured out as their earlier instars over the winter. So you get, if you take the Dicosmicus out of the picture, which we did with experiments that started in mid-late June, you can recover algal biomass, enough to cover these artificial substrates and natural ones that are grazed clean when Dicosmicus is present at normal density. You take them out, you get scuzzed up substrates and modest floating mats, but this is kind of a modest recovery. Still, they're having a lot bigger effect than steelhead. So the experiment is steelhead plus or minus and Dicosmicus plus or minus, and here the results show that steelheads still are a bit bad for algae. There's more algae if steelhead are absent, but the big effect is whether Dicosmicus are present or absent. So you'd have to call this, if you think in cartoon fashion, as I do often, a 2-level food chain. The lack of scour then gives us a two-level food web, and these members are still present, but they're not functionally important. Fish are probably not eating as well. Michael Parker and I did a study to simulate flood drought conditions and found that under the drought simulations, steelhead juveniles didn't grow as well. So this is what you get in drought years, and also where we've done disturbance removal experiments throughout California. You do find a lot more predator-resistant grazers that can build up densities and then make the trophic cul-de-sac to channel all of the algal energy into their populations and not feeding predators as well. Okay, I want to talk more about why algae matter. And I owe a lot of what I know about algae to a third mentor. I've been very lucky to have Tom and Bob in my life, and also Rex Lau, who loves algae, and if you spend more than a day with him, you'll love them too. So here he is standing in a post-flood, nice green Cladophora bloom. Remind you that Cladophora, when it proliferates, it's hugely increasing surface area, maybe 5 orders of magnitude or so. It starts its life out as a green alga. I think of it as Rapunzel's hair, actually, as you can see probably why. And so she starts out with green hair, and then in her middle age, she becomes a blonde. And we're going late into the summer here. Then late age, she becomes a redhead. And the difference is the depth of the diatoms and the species of diatoms that are colonizing these substrates. So diatoms really are God's gift to the natural food web. They are full of fatty acids, amino acids, just really high nutritional value, and they're relatively easy for most grazers to digest because these glass cases that they live in are pretty full of pores and easy to crunch. So here's the yellow, the blonde phase, are diatoms that stick right to the host. This is the Cladophora. Here's the Cladophora and the diatoms. And then a few are starting to protrude up as nutrients or light gets a little bit limiting as life gets crowded down there. But they're all attached to the primary substrate, some with gelatinous stalks, some with their own glue. That's the yellow phase. Later, we get this really key organism in the eel, which is a diatom that's very mobile. It can climb up and over each other's shoulders. And what you're seeing in this electron micrograph is Cladophora that's 5 or 10 cells deep in a particular genus of diatom. These are the same genus, they're 2 different species here. It's epithemia. And Sharon Lawler is in the audience. I was saying epithemia, it's the Greek word for desire. Why should it be called epithemia? And she said, oh, it's because it looks like human lips in some of the, so I thought that was brilliant, stayed with me. Maybe this one here. But so epithemia not only is mobile, but it also has endosymbiotic cyanobacteria in it. People at Davis here have worked out the genomics for a relative and to figure out that these cyanos are very close to one of the picoplankton in the ocean. But here it is in a dead diatom, and these cyanos are not long for this world either because they need the diatom. It's very obligate mutualism. Here, you can barely make them out in a living diatom. They're a little bit easier to see here. So this makes the diatom have a lot of access to nitrogen, which in late summer is getting more limiting because it's been more taken up in the, mainly probably by the green chlorophytes. And that makes them yummy. Now, cyanobacteria are not always going to use that atmospheric nitrogen they fix to make nice stuff. Sometimes they make toxins with it too. But the cyanobacteria here make something delicious because our midges are just full of diatoms. And when it comes out the other end, they're empty diatom shells. 90% of what they're egesting here is diatoms. Also, you can see the midges, while they're making their houses out of Cladophora, are cleaning the neighboring Cladophora of its diatoms because around the tufts, it gets green again. They're greening up their neighborhood by grooming off these epiphytic algae. So it's clear just from macroscopic observations that these are feeding the midges. If you look at emergence, you're getting about 25-fold increase in bug meat coming out of the river and in to feed bats and birds and spiders if you're growing over the rusty Cladophora than over either green Cladophora or other kinds of algae that don't attract epiphytes. And here are a few data on nitrogen fixation, biovolume of epithemia on the cladophora when it's green, yellow, or red, and the amount of nitrogen fixed. And that seems to be pumping nitrogen into the river. Here we have data that Jock collected in June when it's just getting started. These are drainage areas from that 1 square kilometer tiny stream I showed you through to about 100 square kilometers, which is as large as we get, about 150 within the Angelo Reserve. And here it is in June, but here it is in August when the epithemia has really gotten cranking. So there are other sources of nitrogen, like alders, that we've checked, which aren't leaking in the summer. So we're pretty sure that this is mostly cyanobacteria and probably largely the one that lives in epithemia. So now you have these organisms coming and going in the upper watershed. What are their fates? We've done a little work I won't talk about on what happens if an alga gets eaten by an aquatic bug and it emerges and gets eaten by a bat. Or it could have a more inglorious fate, which is it just gets stranded and turns into that white algal paper you see along shorelines, but it could rot, or more often probably it gets grazed by specialized grasshoppers we have and carabid beetles, and there are a lot of little frogs preying on those. So there's a little food web for stranding that's under-investigated and kind of interesting. But it can move up and down in a river fish if the bug gets eaten by the fish, or the algae directly in some cases. Or it could go down the estuary, it could get offshore, and it could go into the deep ocean canyon. And this is in black for a reason. I don't think in my lifetime I'll ever get to investigate the near-shore open ocean, because I can't afford a glider and wouldn't know what to do with one. But the estuary, we'd like to stick our toes in the estuary, and this is very incautious because we don't know much about it, but we want to do it. And then also we've been very lucky with the deep ocean burial as a fate for carbon. And I'll talk about that after I just show you the rest of the eel. Here's the little postage stamp that is the Angelo Reserve. And then here's the South Fork draining down to the main stem and going out a modest delta and floodplain and then into the ocean. And because of a number of things, those great big floods and bad, really poor, brutal logging, we have a wide, shallow river all the way down. There are very deep holes. There are cold water pockets, but a lot of the eel is wadeable all the way down to the mouth. And so you can put these little transect tapes up and try to keep track of the algae. We want to improve our lives by getting into the drone game, obviously. But the phenology of the algae is pretty similar all the way downstream. It's kind of accelerated as it gets warmer and sunnier downstream. And you can see, by the way, looking off the various bridges, that drones would work in terms of picking up that color variation, yellow, green, red, and green. And then we just have to do a lot of muddy boots field work to figure out if that means what we think it means in terms of nitrogen in and bugs out. Charlene Ng was the graduate student who first got us into 30 parts per thousand salinity. She was determined to work in the ocean. And there she is at the very mouth of the eel. The eel, by the way, is a short, steep river for its size, so it stays gravelly almost all the way down to the estuary. And those gravels are just full of crustacea, microcrustacea, amphipods and isopods. And so they're feeding shorebirds and the fish that are spending some time in the estuary. So what Charlene did was to bring river and algae down and compare it as an attractant with the local ulva and enteromorpha that are growing in the estuary. And these amphipods and isopods were very bored by the enteromorpha and ulva. They were very bored by the soft green stuff, but they loved the river stuff. Here are some slides, and Charlene has the time to demolition for these little, what she did, which I thought was extremely clever, is just use these disposable pipettes, cut a slit in them, pinch the algae, and then sit back and watch in the estuary. And they get blackened with amphipods and isopods and disappear within minutes if they're river algae. So, and they don't. These are boring. And the reason you see that soft green algae there is because it's not being eaten. So the fun hypothesis that we want to test. And there are complexities. We are very eager to work with a lot of UC Davis folks, as you can imagine, on these complexities. This depends on tidal height, river conditions. You know, a lot of these things are going to be influencing this interaction, but at least for some parts of the year, we think this river carbon is a hidden cryptic carbon source that needs to be accounted for, not in carbon burial budgets, but in trophic budgets for the estuary. So how do you know how much carbon's coming in from river algae if it disappears right away? And so our approach was typically sophisticated. We just got a big net. And Charlene found one miracle cross-section just below the Fern Bridge, for those of you who know the eel, where you could stretch a net almost all the way across. Even at the lowest flow, the river's moving too fast to keep a seine down for certain parts of the river. But Charlene, even though she's small in stature, is very good at bossing big, strong people around. So she got most of it walled off. And then for the challenging parts, we had our stalwart Berkeley undergrads and grad students. This is Will Torres and Charles Post, holding frames, big box frames that Charlene had built that were stiff in place. And even if they got pushed back 10 meters during the 10-minute sampling, they still maintained the cross-section. So we got the numbers, so that was great. It was great fun, and I think it wasn't terribly dangerous, but probably a little dangerous. So it certainly got dangerous in November, but then we subsampled. So the algae starts moving from upriver through the Fern Bridge transect we did was about the highest point where it was freshwater, even at the highest tides and lowest flows. So that's where it's entering what we would consider the brackish estuary. And there's a little brown thing. I kept it on arithmetic because I think that tells the story more clearly, but there's a little bit of leaf litter moving out, but you can't even see it on this graph until the first bankful flood happens. The bankful flood is enough water to inundate the winter wet but summer dry banks. And that's where all the forest leaf litter and vegetation is stored. So it only moves in one big pulse out in November, and there's more at that point than the remnant algae. But this stuff is no good to any food web, river or estuary, because it's moving out too fast. And this is probably what's important, though, in sequestering carbon from our river and rivers like it, because it moves out, gets into the deep canyon, gets hopefully buried, and takes it away from the atmosphere, we hope. However, this is the stuff that matters probably to those marine food webs, and we don't know how much yet, but we're interested. So you've got then these scales, and we hope for, again, put the plug in, a lot of help from the marine and estuarine people at Davis, and it's been fun to hatch plans already. But this is off limits, I think, with our budgets and expertise. This was not off limits because an oceanographer at University of Washington, Chuck Nittrower, I don't know if any of you know him, did he did cores in the mouth of the eel. Here's the eel mouth. This is a big sediment plume that's entering a place where the Eel River has carved big canyons, deep canyons like those off Monterey Bay. And so about 50% or more of the eel's sediment every year ends up in those canyons depositing. And Chuck collected about 50 sediment cores from there. I'm very interested in sediment transport dynamics. He's a marine geophysicist at UW, University of Washington. So here we are at sea looking back at the Coast Range, vertically exaggerated. Here's some branches of those canyons and the material that they're coring into with an MBARI-funded remote vehicle. And the cores come up looking like that. Of about 90 cores that he collected, only 3 were laid down with enough flatness and evenness that they could be annually dated with his lead 210 dating. But there were 3. And I asked him if they'd ever looked for diatoms in those cores, and he said no, and he gave us half of some of his very good cores. And so Jack Scully was able to do a dissertation looking for freshwater diatoms that looked totally different than marine diatoms in these cores. They named the canyons, by the way, Larry, Shemp, Curly, and Mo. And our cores all came from Shemp, in case you're interested. So here are the data that allow us to date the cores by about 11 millimeter increments and then 6 millimeter increments. And the change in slope between age and radioactivity here, there's a lot more sediment deposited with every event after the river was destabilized in the 64 event. So now every storm scours in much more bankside sediments, and that's showing up in the record. But still, you have a nice linear relationship that lets you look at the cores, see how old, try to find a year that corresponds to modern years, and then look for the diatoms. So epithemia has a very characteristic shape, and this is a modern cell, but here are the cells that Jack is pulling out of those cores. And the epithemia is a robust frustral, so our friend Rex Lau told us that they will preserve the best. You can see cochineus, which is the flat disc, is starting to dissolve even after 83 years. And here are some of the other freshwater diatoms that have shown up too. They don't look anything like the centric diatoms that are coming down from the ocean. I should also mention that this core gives us 83 years because the 1906 earthquake shook away the record, and then it took about 12 years for the sediments to even out so that they would start accumulating in a datable fashion. What was really unexpected but pretty useful is that the frustral density in those cores, which would be the total, we're using that as our predictor value now, because can you tell what the river upstream was doing from the density of frustrals in a sediment layer dated to a certain year? And well, if you believe in R-squareds of 33 or 47, yes, you can get a good idea. We were actually really surprised it came out this well, because there's actually quite a bit of noise between discharge and algal abundance. Even in the modern record, sometimes you get a low algae year with a high discharge, and it's because you get a secondary spate that scours out the algae after it starts growing, and then the dune conditions aren't very good to regrow. So you can get noise from that record, but that noise actually was useful to Jack Scully because he could then tease apart, is it really a proxy for productivity of the algae, or is it just related to sediment or discharge? So he could tease that apart, and it was better related to the actual productivity that I had measured by doing lots of these tedious upstream transects of my record that started in 1988 and is still going. Only at the time, 13 years overlapped with Chuck's record, which was collected in 2001. So we'd like to collect another core if we could figure out how to fund the operation and see what's happening, because we have a lot more points we could put on to test this. But if you buy into this, then you can go back 83 years and think about big algae years and low algae years and revisit this question of, are these algae limited by top-down grazer extirpation or bottom-up, prolonging good growth conditions, effects of floods? So which is going on at this whole basin scale? And here are data that Jack Scully got from the rainfall records that go back a long ways, back to the beginning of the record. I think these are from Ukiah. You got them from Eureka. There are a couple of stations. This is Eureka. All of the weather records are saying the same thing, that the total precipitation that year has a noisy, and if you want to read into too many points, a negative relationship with algae. Well, that's kind of surprising, but maybe it was just darkness. Maybe there was too much scour after growth. Something is not making wet years better for Cladophora. So it looks like that leaves the top-down hypothesis as the major explanation. And so just to quickly sum up, since I'm out of time, the idea is that if you want to produce salmon, or if you just care about the welfare of other things in the food web, you would like to have a scouring winter to get rid of the big grazers, and then normal summer flows with high enough space flow to really flush the stream. If you don't get that, you'll get the food sequestered into inedible grazers, but there's still a lot of epiphytic production, so the salmon can still probably make some growth. And here's the last food web that we're really worried about. It's become recently a big concern is what happens if you have either kind of winter and then an extraordinarily dry summer where the base flow is way too low and even the main stems can get warm and somewhat stagnant. And how can you get a dry summer following a wet winter? And this is the answer. You have enough marijuana growth in a lot of our watersheds, indoor and outdoor grows, to dry up large tributaries. So that's what's happening, and that's what needs that and all the other problems of marijuana, which you guys at Davis are making huge contributions to tracking and figuring those out. But there are those problems, and then you can also just add a lot more sediment and completely reverse a lot of the recovery and restoration that has helped the river recover from the devastation of bad logging in the '64 flood. So the green rush is very, very bad for, of course, North Coast rivers, and we'll be very interested to see what happens under the legalization regime. Here's the very worst case. You get one scouring flood in the winter before, then it's sunny and 70, and the cladophora gets an early start and gets massive. This is the biggest cladophora year I've ever seen. This is 2012 to 2013. And then you have a little spate in June that takes the 14-foot-long Rapunzel's hair and jerks it loose, but there's not enough water to push it out to the ocean, so it stays in place and rots. And so these are the floating mats. And what's going on is these diatoms, which now that you know and love diatoms, you know they should be a healthy orange color. These are turning green because they've lost their carotenoids and they're bleeding their chlorophyll into the matrix. And who's there to suck it up but cyanobacteria? So Hans Perl has done a lot of useful work on predicting that in a warmer world we'll have more cyanobacterial blooms because they like it hot. These are growth rates, this is temperature. Diatoms like it colder and peak out around low 20s. Greens, mid-20s. Cyanos are going strong towards into the 30s and probably into the 40s. And that's what these mats do. They elevate temperature. You put a thermistor in the mats, it moves up into the mid-30s, whereas just a few centimeters in the water, it's stabilized to a more tolerable temperature. But I hope you can empathize now with the dying diatom. That's one of my goals. And now feel goosebumps as you see the evil Anabina unwinding here, furling up. They're in these mats, probably coiled up like earthworms to protect themselves from UV when they're up there, but they're soaking up nutrients. And then they start colonizing these rusty red, good cladophora diatom assemblages. And first it's few specks of blue-green, but then it's a cloak. So here is a margin between rusty red algae and vegetatively spreading Anabina, which is one of the neurotoxic ones. Underwater, it looks like this. I'll show you another picture that in a moment that will explain this, but they're just little forests of cloaked over standing algae that are now draped with a neurotoxic cyanobacteria. Here's a better picture that shows the original good algae. It was infected, this thing spread, gathered bubbles, and then this lifts off, spreads, collects in areas where dogs jump in the wrong backwater, lick their fur, and die in convulsions in front of their owners in sometimes 20 or 30 minutes. And kids are at risk at places like this. So this, the natural history of these cyanobacteria in rivers is very, very poorly known. It's wide open. We don't know how these things spread, how they overwinter. We know a little bit more maybe about their thresholds for growth. We don't, now in the era of genomics, you can do genomics. Keith is working on another neurotoxic algae that actually grows attached. We first see Anabina as it is collected on cladophora meshes. So we don't know where it comes from, but these, we know where they're growing throughout the watershed. So he's using techniques like this to transfer them around and then sequencing the whole thing and trying to make sense out of the Indian blanket that is the genome and the co-occurring heterotrophic bacteria. It's very, very complicated, not straightforward, but a nice open frontier. Here's a crude map of dog deaths. Here's Keith training folks how to recognize the algae they need to thank from the algae they need to fear, collecting with Raf Kudala's help cyanotoxins and following that over the eel. And the last message is kind of what are rivers going to deliver to the ocean? Are they going to deliver wonderful edible diatoms, or are they going to deliver cyanotoxins? In the Monterey Bay, Rafe Kudala and Melissa Miller and others have discovered up to 20 dead sea otters died of microcystin liver toxins, and those had to be produced in the agriculturally enriched rivers. So we've got to be careful what we're sending out to sea. And how much and what we're sending out depends on the little biogeochemical factories that are these little algal assemblages and methylating mercury, fixing nitrogen, denitrifying. All this magic is happening on Cladophora and other substrates, of course, but how much of that's going on depends on how big the Cladophora is and where on the gradient of anoxic crotches to well-flushed tips and well-lit tips it's growing. There's all sorts of interesting stuff there, but how much of this proliferates depends on what the food web's doing to it. What the food web's doing to it depends in part on where it grew, but also, of course, what winter and summer hydrology did to it. So sending out yummy stuff or yucky stuff to the ocean. And one of the most fun things about this for us is that it's really made us make friends with our neighbors, because this has gotten their attention. Rex Lau, Paula Fury, and I host, and they teach a wonderful algal identification course. The locals learn to actually really love the algae and recognize the ones they need to keep their dogs away from. And so that's been great fun. We were even hosted by the Yurok tribe the last time we taught this in 2017, so that was great. So I just want to end with saying the one thing we individuals and small local groups can do, of course, is take care of our watersheds and take care of our subsurface. So there's this network of what's called critical zone observatories, I think I'll end with this slide, that is paying a lot of attention. One's at Angelo, one's in Yosemite in Southern California, and then there are 7 others distributed through the US and Puerto Rico. This is a program that looks at the water exchange from what's under our feet down to where it doesn't exchange any longer, the unweathered bedrock. This feeds rivers after it stops raining, and this feeds forests. The forests are getting a lot of their moisture from the vadose zone that's not saturated. So these things are critically important to not only understand, but to take care of. So if you get involved in any environmental issues, I think we can shake our fists at big government and maybe vote correctly, but here's one that you can do. You can really are empowered to think about at scales that we can control. So with that, thank you for letting me go 9 minutes over, sorry, and would welcome questions. Peter, yeah.
Cladophora, I guess I'm wondering what is it about it that's hard for things to eat? What makes it hard for fish or insects to eat it? And then I'm also wondering if as you go south along the coast, if eventually you pick up fish taxa, for example, in the tropics or subtropics that actually can eat it. And if that,
I think Cladophora is edible, but it has a thick cellulose cell wall, so it's probably not quite as digestible, but a lot of things do eat it. I think it's just the cracker, and they'd like the peanut butter, which are the diatoms. That's the way stream ecologists have always metaphorized that. Yeah. Yeah.
So clearly all these food webs are very dynamic and dependent upon different situations. And what do we know about these food webs and how they might behave in dammed systems? So many of our rivers are subject to that.
The little bit of work I've done, dams do 3 bad things in California, or where you have salmonas, but it's well known that you run out of spawning gravels downstream from dams. You also armor the bed, and so you can't cleanse the hyporheic and get pore spaces that keep that ecosystem healthy and give prey species a little bit of a refuge so that they can maintain themselves. But the main thing is it encourages invasive species, and you at Davis have done a lot of the work on that. The part in the food web is you get more predator-resistant grazers downstream from dams because you haven't obliterated and reset the food web in a way that rejuvenates it. So when you scour downstream, if you could scour that, you'd reset succession, and according to RK selection theory, you'd get fast-growing edible things, and that's often what we see, midges and mayflies, that are good fish food. And it's only later, even towards the end of a post-scour summer, you start to see buildup of predator-resistant grazers because the others have been gleaned out. But so that rejuvenating the food web, so there are three things you can think of: invasives, sediment clogging and disruption, and rejuvenating the food web are all what dams do. The management solution is maybe just one good scour would do it. But yeah. Yes?
How do you think the 64 flood and the increase in sediment load affects the kind of annual reset when you do have a flood as far as scour and impeccability?
It's probably much worse and much more unstable. When we in the areas, we've actually, in the 40 years since that bad logging, we've seen recovery, and the riparian alder groves have grown to diameters of about 40 centimeters, and they can really not only stop a lot of the bank erosion, but they can start to divide the river so it gets deeper and narrower, and of course more shaded. So that's a more stabilized, that's probably working down to about maybe 200 square kilometer drainages, and then it would get too wide for the alders to have that kind of effect. But it's very useful in keeping the water backed up, and then you're getting more groundwater, it'll be colder, and you're getting the shade, which is another big part of the temperature, and you'll have more complex cover. So if you want a cold water fauna, you better recover the alders where you can, or the willows. But so the loss of, under the widening and shallowing and terrible bank erosion, we lost the riparian galleries, which is huge. And that's perhaps one of the reasons the river went to ground is it's not necessarily there wasn't enough water. It might be that the water was undergrounded by too much fine sediment.
Thank you. All right, again.
Great, now we get to party.